Hi children, good morning to you all. How are you all? Hope all are doing good and praying to be good. So happy to see you back again through this session, and I come back with a new session or a new chapter. So in your previous class we discussed the fundamental unit of life. Yes? What is the fundamental unit of life? Cell is the fundamental unit of life. So why it is called as fundamental unit of life? Because all the living organisms are made by the cell, and without cell no organism is formed. So and the cell consists of many cell organelles. Each and every cell organelle, it has its function to activate the life process of a cell. So cell is known as the fundamental unit of life. So how the organisms are formed, children? Organisms are formed from a group of cells which are structurally functionally same. They develop in the form of a tissue. So a group of tissues structurally functionally same, they develop to form a organ. A group of organs, they develop the organ system. A group of organ systems helps to form the organism. So in this way the organisms are formed. So cell is the basic unit of a life. So if the cell has to work effectively, it needs energy. If we want to do any work, we need the energy. How we used to get the energy? Through the food. Yes or no? By taking the food, we used to get the energy. So how the cell get the energy? By the food. So as soon as we took the food, do we get energy? No, it has to be digested, it has to be mixed with the blood, then it has to mixed with the oxygen, then the energy has to be liberated. Yes or no? So many processes take place to produce the energy in the organism. So if an organism has to survive, it needs to produce energy. If the energy has to produce, a lot of process has to be takes place. So the processes which are taking place in an organism to survive the life is called as life process. So in our 10th class, the first chapter of your biology, we are going to learn about life process. What is a life process? Life means which are able to live, which are having a life and which are able to continue the life. They are called as that is called as a life. In order to continue the life, many activities has to take place in the body. So any action takes place in order manner, that is called as a process. To have a better life, we used to have the processes, many processes. So the processes which support the life of an organism are called as life process. So in this chapter, we are going to learn about the different types of life processes. So how the life processes are helpful for the survival of an organism. So coming to life process, biology is the science that deals with the living things. Sometimes it is necessary to make a difference between organisms that are living and other things and that are alive, not alive. So if you want to see the difference, biology always deals with bio means life. Logos means study. The study of life is nothing but is a biology. It always interact or the study. Biology always helps to study about the living organisms. Okay, so it helps to study it is a science. Helps to study about the living organisms. When there is a living organisms, the living organisms cannot survive without non-living organisms. Yes or no? So there is always an interaction between the living things and the non-living things. First let us see the characteristics of living things. How can we define what are the living things and what are non-living things? So living things always react to stimuli. So if I call you by your name, you will respond. For example, I call you Anand or Santosh, something. If I call the name, if the person is there, they will react. If I call hi stone, that the stone can respond? No. Hi wall, hi board, hi chalk. If I call, that things can respond to me? No, because they doesn't have any ability to respond. They doesn't have any response to stimulus. What is a stimuli? If any action takes place on the body, that condition is called as a stimuli. So living things always interact with their environment. We used to interact with our environment. We interact with the air, we interact with the water, we interact with the soil, we interact with everything. So the living things has a capacity to interact with the environment, which includes members of the same and other species too. So we doesn't interact only man to man. We have an interaction with the birds, we have an interaction with the animals, we have an interaction with the snakes, we have an interaction with the different kinds of animals. No? So we doesn't interact only ourselves. We used to interact. The living things always interact with the from species to another species. That is also one of the character of living things. And living things have metabolism. What is a metabolism? Energy releasing processes, able to produce energy by making different processes. That is called as energy releasing, is nothing but is a metabolism. These metabolic activities can be seen only in the living organisms, where we doesn't find in the non-living things. So they take in food and which they convert to the energy they need. So they used to maintain the metabolic processes. Due to the metabolic processes, they produce the energy. So the food is which we eat, it helps to ultimately produce the energy. So living things produce, they give birth to others of the same species. This is not true of all individual organisms. In a eusocial organism, some castes cannot reproduce, but since they sterile, workers are all the producers of a single queen. They are one collective. So if you take a honeybees, in honeybees, all the honeybees cannot reproduce. Yes or no? The same manner, maximum the characteristic of ability to produce is one of the characteristic which defines or decides the characteristic of a living organism. So these are some of the characteristics to define that what are the living organisms. Ability of stimulation, interaction with the environment and other species, and making of metabolic activities which helps to produce energy, ability to reproduce. These all can be considered as the process of organisms which are having the life, living organisms. So let us define the life process. Various functions carried out by living beings which are necessary to maintain the continued life are called as life process. So various functions are carried out by the living beings which are necessary to maintain a continued life are called as life process. So to continue the life, number of activities, number of functions will take place in an organism, and these activities or these functions ability to continue the life. So following are the life process in a living beings. What are the life processes in the living beings? Nutrition, respiration, transportation of substances, excretion, movement, reproduction. What are they? Nutrition, respiration, transportation of substances, excretion, movement and reproduction. These all are the activities of living beings. So let us see characteristics of living things. Some characters already we discussed. Again we'll see. That is movement, respiration, sensitivity, growth, reproduction, excretion, nutrition. All these activities can be seen in only a living things. We cannot see in the non-living things. Movement, respiration, sensitivity, growth, reproduction and excretion. These are the characteristics which can be seen only in living organisms. So let us discuss about the process one by one. Okay. So coming to the nutrition, let's go in the detail of a explanation of the nutrition. Let us see about the nutrition. So the process by which an organism takes food and utilize is called as nutrition. So the process of taking food and it is utilized in the form of energy is called as a nutrition. And why the nutrition is needed? nutrition. Organisms need energy to perform various activities. The energy is supplied by the nutrients. Organisms need various raw materials for growth and repair. These raw materials are provided by the nutrients. All the raw materials are provided by the nutrients. So if the organism has to grow, if the organism has to develop, they need the nutrition. What is the nutrition? The process of taking a food and it is utilizing in the form of energy to all the organs is nothing but is called as the nutrition. So we used to take different types of the nutrients. What are the nutrients? Materials which provide nutrition to organisms are called as nutrients. The materials which provide nutrients like that, carbohydrates, proteins and fats are the main nutrients and are called as macronutrients. Carbohydrates, proteins, fats. These are the main nutrients. As they are main nutrients, we can call them as the macronutrients. So macronutrients, minerals and vitamins are required in a small amounts. Minerals, vitamins, even fibers and even the water, they are needed as a minute components. So small quantities. So they are required in a small quantities. They are called as micronutrients. Minerals and vitamins required in a small amount. So they are called as a micronutrients. So basing on intaking of food, the organisms are taking different types of foods. Basing on the type of food, the accepting of food, the process of nutrition is of two types. That is autotrophic nutrition and heterotrophic nutrition. Autotrophic nutrition and heterotrophic nutrition. First, let us see what is meant by autotrophic nutrition. Autotrophic, auto means self, okay, making by ourselves, organisms which they could able to prepare their food by themselves. Such a type of nutrition is called as autotrophic nutrition. The mode of nutrition which an organism prepare its own food is called autotrophic nutrition. Green plants and blue-green algae follow the autotrophic mode of nutrition. What are the examples? Green plants, all the green plants and the blue-green algae, these can prepare their food. Organisms which can prepare their own food by themselves are called as autotrophic nutrition. So, we think we have forgot something, no? How the plants used to take the food. Yes or no? How they can prepare? Why we cannot prepare the food substances? How the other organisms able to take a food? Yes, we know that. The plants used to prepare the food by the process of photosynthesis. So, the plants are capable to prepare the food by the process of photosynthesis, and animals cannot synthesize the food substances. They cannot prepare their food by themselves. So, let us see what is meant by photosynthesis. So, conversion of the food substances into light energy and normally from the sun into the chemical energy that can be later released to the fuel, the organism activities. So, the plants used to use the sunlight and prepare the food substances. So, these chemical energy is stored in carbohydrate molecules such as sugars, which are synthesized from the carbon dioxide and water. So, in most cases, oxygen is also released as a waste product. So, when the plants prepare their food substances, they convert the carbon dioxide into carbohydrates by making use of the sunlight. So, in this case, during the process of photosynthesis, the carbon dioxide eliminated by the plants as the waste substance, unnecessary product. So photosynthesis maintain the atmospheric level of oxygen content. So it helps to maintain the balancing of the atmospheric temperature or the light condition. So what happened during the photosynthesis? So I know that, go to next slide to see the answer. So that fellow want to see the answer. So let us see what happened during the process of photosynthesis. Let us see to the next slide. Light energy is absorbed by a chlorophyll, a green substance found in the chloroplast in some plants, cells and algae. So the plants are consisting of chlorophyll. The chloroplast are special cells which are called as a chloroplast. Already we discussed in our ninth class, no? So chloroplast cell. These chloroplast cell, they contain a pigment called chlorophyll. So this chlorophyll helps to absorb the light energy and used to convert the carbon dioxide from the air and water from the soil into the sugar called as a glucose. Only this chloroplast cells, they contain a pigment called as a chlorophyll. This chlorophyll able to absorb the solar energy and convert the solar energy into chemical energy, and it make use of the carbon dioxide into carbohydrates. So oxygen, so these during this process the oxygen is released as the byproduct. It will be released as a byproduct. So what is the importance, what is the significance of this photosynthesis? Photosynthesis is the main way through which the solar energy is made available for different living beings. The solar energy, in the energy itself we cannot make use of the solar energy, but the photosynthesis is the main process which make us to use the solar energy in the form of food sources. So green plants are the main producers of a food in the ecosystem, in the environment. All other organisms directly or indirectly depend on the green plants for the food. So all other organisms, they will directly or indirectly, they will depend on the green plants for their food. So the process of photosynthesis also helps in maintaining the balance of carbon dioxide and oxygen in the air. So what happened? This photosynthesis always helps to, so the increasing of carbon dioxide in the environment is made use by the plants, and their carbon dioxide will converts to the food for the ultimate population in the living world. So we can say that this process of photosynthesis is helping to balancing the carbon dioxide level. So how this carbon dioxide is entering into the plant body? How this is so the plants are having special structures which are called as stomata. We can find at the base of the leaf, the porous like a structure is called as a stomata. A pore found in the epidermal of a leaf, stem and other organs that is used to control the gaseous exchange, mainly the gaseous exchange. In which activities we used to find the gaseous exchange? We find in the process of respiration and process of photosynthesis. In the process of photosynthesis, plants take inside the carbon dioxide, they leave the oxygen. In case of respiration, the plants take inside the oxygen and they leave out of the carbon dioxide. So this is nothing but it is a gaseous exchange process, changing, taking in and leaving out, giving out something and taking inside another one. That is nothing but exchange. Gaseous exchange, the process of gaseous exchange is of two types in plants, that is a respiration and a photosynthesis. So these two processes are controlled by the stomata. A pore found in epidermis of a leaf, stem and other organs that is used to control the gaseous exchange. The pore is bordered by a pair of specialized parenchyma cells known as guard cells. So the stomata consists of outermost border. They have a special cells that are called as a guard cells that are responsible for regulating the size of opening and closing of the stomata. So the availability of water, basing on the availability of the water and the temperature, the opening and closing of stomata. These guard cells, they are lives at the both sides of the stomata. By opening and closing, if the guard cells helps to open and close, when they are opening, the gaseous will be enter and excess water will be eliminated of the plant body. So the mechanism of closing and opening of stomata is always controlled by the guard cells which are on either sides of the stomata. So the term is also used collectively to refer to an entire stomatal complex, both pore itself and it is accompanying in guard cells. So on either sides the guard cells are present and they support the activity of the pores. So air containing carbon dioxide and oxygen enters the plant through this opening and is used in process of photosynthesis. So mesophils, parenchyma cells, which chloroplast and respiration respectively. So the leaf contains a special tissue that we call it as a mesophil tissue. This mesophil tissue is made by the parenchyma. So parenchyma, when we studied in your 9th class tissues, parenchyma have two types of cells, parenchyma and chlorenchyma. This chlorenchyma is located in the mesophyll. So it is consists of rich content of chlorophyll and which helps to trap the solar energy and maintain the process of photosynthesis. Oxygen produced as byproduct of photosynthesis diffusing out the atmosphere through these some openings. So the oxygen is a byproduct of the process of photosynthesis in the plants. So also water vapor is released into the atmosphere through these pores in a process called as transpiration. So the excessive amount of the water is released by the pores of the stomata. The process of releasing excessive amount of water through the pores of stomata is called as a, that we call it as a transpiration process. Let us see what is meant by heterotrophic nutrition. Heterotrophic nutrition, it is the mode of a nutrition which organisms takes food from another organisms is called as heterotrophic. So hetero means others, hetero. Auto means self, hetero means others. Depending on others and taking the food from others is nothing but is called such type of nutrition is called as a heterotrophic nutrition. So organisms other than green plants and blue-green algae flow heterotrophic mode of nutrition. So other than autotrophs. What are autotrophs we discussed? The plants and the blue-green algae. All the green plants and the blue-green algae, they are autotrophs. Other than these two, all the other living organisms, they show the mode of heterotrophic nutrition. So heterotrophic nutrition can be further divided into three types. So that is saprophytic nutrition, holozoic nutrition and parasitic nutrition. So what are the three types of nutrition, children? Classification of heterotrophic nutrition, saprophytic nutrition, holozoic nutrition and parasitic nutrition. Let us have discussion detailedly what is meant by the saprophytic nutrition. In saprophytic nutrition, the digestion of food takes place before ingestion of a food. So generally after eating the food is digested. But in saprophytic nutrition, first the food will be dissolved or digested. The digested food or the dissolved food will be taken by the organisms. So this type of nutrition is usually seen in fungi and some other microorganisms. We can see in bacteria, in fungus, some other microorganisms we can find. The organism secretes digestive enzymes on the food and then ingested the simple substances. So saprophytes, what they used to do, they release when they are attaches to the on the surface, when they reaches to the substratum of the food substance, they release enzymes into the food and they dissolve the food substance and they digest the food substance and they convert in the form of a liquid, simple form substances. So saprophytes feed on dead materials, help in decomposition and remains of the plants and animals. So they used to mostly they act on the dead and decaying material. You can see here mushrooms. These are the fungus. They used to grow on dead plants, no? Most of the dead plants. So the fungus and the bacteria, they used to adjust on the dead materials and they convert the dead materials on the decaying condition. How they cause the decaying? By releasing the enzymes into the substances and they cause the decaying of the substances and they used to make out. Now let us see what is a saprophytic nutrition. This nutrition takes place before first digestion and then intaking of food is happen. The fungus and the bacteria, they release the enzymes into the food substances and they dissolve or dilute the food substances, convert into a simple form and they'll absorb the food. Such a nutrition is called as saprophytic nutrition. Sap means is a liquid. So solid food, by releasing the enzymes, it will convert in the form of a sap. This sap is eaten by the organisms. So that are called as saprophytic nutrition. Example, we can take a fungus and the bacteria. Now the second one is a holozoic nutrition. What is a holozoic nutrition? So in holozoic nutrition, the digestion of food follows after ingestion of food. Thus digestion takes place inside the body of an organism. Holozoic nutrition happens in 5 steps, that is ingestion, digestion, absorption, assimilation and digestion. Ingestion, digestion, absorption, assimilation and digestion. Generally, in saprophytic nutrition, we see that first digestion over, then ingestion will take place. What is an ingestion? The process of inputting the food into the mouth or into the body. Sending the food into the body or into the organism is nothing but we call it as an ingestion. In saprophytic nutrition, first it happens the process of digestion, breaking down of complex substances into simple substances with the help of a digestive enzymes. So here we can see that ingestion of food is taking place in holozoic nutrition. The first step is ingestion, next is digestion, next absorption, absorbing the digested food, the digested nutrients absorbed into the blood, and assimilation, spreading over to all the organs, and egestion. Egestion means excretion, elimination of out of the body. So let us see one step by one. First one, steps involved in the holozoic nutrition. What is meant by ingestion? Ingestion is the process of taking complex organic food through the mouth opening. So through the mouth opening, we are taking complex. Complex food means, for example, rice is a complex content, idli is a complex, dosa is a complex, bonda, bhaji, punuku. These all foods are the complex content. We used to take them into the body, but after the digestion, they will convert in the form of a liquid, very simple form. Okay, so complex substances taking into the mouth through the opening of the mouth is called as a ingestion. Next, what is meant by digestion? Changing of complex food. Complex means somewhat heavy substance. Complex food into simple forms by the action of enzymes. Acting, the digestive enzymes acts on the food substance. They break down into simple forms, small pieces, and convert them into a food. That is called as a digestion. And next one is absorption. Passing of simple soluble nutrients through the blood or a lymph. After the digestion, the digested food will absorb into the blood. So it will be passed through the blood or through the lymph to all parts of the body. That process is called as absorption. Assimilation means utilization of absorbed food for various metabolic activities. In our body, all the activities, even for a respiration we need energy, for eating also we need energy, for talking we need energy, for writing, for speaking, for everything we need to do energy. So the food which is absorbed into the blood, it will be utilized by all the organs. It will supply through the blood to various organs and utilized for the metabolic processes. Metabolic processes means digestion, respiration. The energy releasing processes are called as a metabolic activities. They are produced to all the organs in the body. Next one is addition. Expelling out of undigested food. Expelling out means sending out, deleting, eliminating the unwanted food substances to the other organisms. That is called as a addition. So the next one is types of holozoic nutrition. What are the different types of holozoic nutrition? Herbivores, carnivores, omnivores. 3 types of organisms are there basing on holozoic nutrition, taking off for different types. So that animals which feed only on the plants, that animals are called as herbivorous animals, and the organisms that feed on other organisms, they are called as carnivores animals. That means only the flesh eating animals, only they eat the flesh, that are called as omnivores. Carnivores, and omnivores, the animals which feed on both animals and plants. They used to feed both on the plants and the animals. Such a type of nutrition is called as omnivorous animals. You can see different examples of holozoic nutrition. You can see lion, tiger, giraffe, ostrich. Different birds are there, animals are there. These all are the animals, different types of animals show that is 3 types of holozoic nutrition. That is a herbivorous, carnivorous, and a omnivorous nutrition. So let us see how the nutrition will take place in amoeba. So amoeba is a unicellular animal which follows the holozoic mode of nutrition. So the cell membrane of a amoeba takes place on producing into pseudopodium. So it is a single cell animal. only one cell has to perform all the activities. So it used to protrude the muscles out of the body. These are the muscles. It used to protrude the muscles out of the body. These protruded muscles come and they grab the food substances out of the surface of the area and they'll take the food into the body. Okay. So amoeba surrounds a food particle which pseudopodia and makes the food vacuole. So it enters into the food vacuole. It develops the vacuole in its body. As it is a single cell, so no different organs will be there, no tissues are there, no organs also will be there. So it forms as a vacuole and it injects the food material into the vacuole. So the food vacuole contains the food particles and water. So the vacuole, what does the vacuole contains? Food particles and water. And digestive enzymes are secreted in the food vacuole and digestion takes place. After food enters into the vacuole, in the vacuole, the digestive enzymes will be secreted. So when the digestive enzymes are secreted, these enzymes facilitate the process of digestion. So the process of digestion will takes place. After that, digestion food is absorbed from the food vacuole. So the digestion completely will take place in the vacuole. After the digestion, it will be spread over to the all parts of the body. So finally the food vacuole moves near the cell membrane and undigested food is expelled out. So when the food is spread over to all parts of the body, the unnecessary food, unwanted food will be spreaded towards the cell membrane. So by the cell membrane again exocytosis will take place. So the food will be comes out. You can observe the picture here, amoeba. So you can see the green structure is a vacuole. The green structure is a nucleus and the brown structure is a vacuole. And the blue color finger-like projections are called as a pseudopodia. The pseudopodia collect the food substances. You can see the picture, contractile vacuoles. So the contractile vacuoles and the food vacuoles, they retake the food into the contractile vacuoles. Due to the vigorous contraction and relaxation and also due to the secretion of digestive enzymes, the food gets to digested. So the process of digestion in the amoeba is also called as a phagocytosis. The process of digestion, phagocytosis. So engulfing the materials into the cell is nothing but we call it as a phagocytosis. Also we call it endocytosis. So the process of nutrition in amoeba we can call as phagocytosis or also we can call as a endocytosis. And the next third type of nutrition is parasitic nutrition. Para means depending on others. Okay, parallelly they are depending on others is nothing but parasitic nutrition. What is a parasitic nutrition? Parasitic nutrition is a mode of heterotrophic nutrition where an organism lives on the body surface or inside the body of other type of organisms. So they live another. For example, this is one worm. This is one body or one cell. It is living inside the cell. So this is called as a host. The organism which giving a shelter to the organism is called as a host. So organisms they live in another organism cause harm. Such type of nutrition is called as parasitic nutrition. The parasitic obtains nutrition directly from the body of the host. So since these parasites derives their nutrients from their host, they'll take the nutrients from their host. So this is symbiotic interaction of often described as the harmful to the host. Sometimes it is a symbiotic. Symbiotic means mutual understanding. If you take root nodules, Rhizobium leguminosarum, it is a bacteria lives in the groundnut roots, no? So this bacteria takes the nutrients from the root and the bacteria convert the nitrogen into nitrites and nitrates, nitrogen fixing bacteria. So both mutually the nitrates are needed to the plant, the root nodules food is needed to the bacteria, mutually exchanging the food substances. No? Parasites are dependent on their host for the survival since host provides nutrition and protection. So the parasites are dependent on the host. They always depend on the host for their survival. So the host provide the nutrition for the protection of these parasites. So as a result of the dependence of parasites have considerable modification to optimize the parasitic nutrition, then therefore their survival. So the parasites, in order to have their survival, they used to depend on other organisms. So parasites are divided into two groups, endoparasites and ectoparasites. Endoparasites are the parasites that live inside the body of the host, are called as endoparasites. Ectoparasites means are the parasites that live on outer surface of the host and generally attaches themselves during the feeding. They will suck the food. Okay, due to the different strategies of the endoparasites and ectoparasites, they require different adaptations in order to acquire nutrients from their host. So they have their own habituations are there, adaptations are there, how to take the food. Basing on their habituations, they used to take up the food from the parasites, from the host. The parasite take the food from the host. You can see examples of parasitic nutrition. Cuscuta, it is an endoparasite. Endoparasite means it entwines to the plant, the roots. The cuscuta is a plant which does not have the chlorophyll. So you can see the white color thread-like structures. They are the cuscuta plants. So they attaches to the green plants. As they don't have a chlorophyll, they cannot prepare the food substance. So they have the haustoria, root-like structures which are called as a haustoria. These roots, this haustoria injects into the plants, host plant, and they suck the nutrients from the host. They'll take the food. So they are standing out of the plant, out of the body or out of the host. Such a nutrition is called as endoparasitic. Mosquitoes, ectoparasites. See what they used to do? They carry the malarial parasite. So they carry the parasites, and what they used to do when they release their saliva, when they bite us, their saliva released into the blood, and they cause the fever for us. No? So enter into the body. That parasites are entering into the body and they are causing the harm to the organisms. So this is well and good how the nutrition is taking place. Now let us see how the nutrition in human beings, which type of nutrition is taking place in the human beings. Let us have the discussion. So human beings are complex animals which have complex digestive system. So the human digestive system is composed of an alimentary canal and some accessory glands. The alimentary canal is divided into several parts, likewise esophagus, stomach, small intestine, large intestine, rectum and anus. Salivary glands, liver, pancreas are the accessory glands which lies outside the alimentary canal. So the glands and the parts, many glands are there in the digestive system to digest the process of human digestion, as well as many organs are there to facilitate better digestion in the human beings. So whatever it may be, the human digestive system is also complex digestive system. First, let us see autotrophic nutrition, how the process of photosynthesis will take place. So, the basic photosynthesis. Photosynthesis is a physico-chemical process. This is a physico-chemical process by which green plants use light energy, solar energies to synthesize the organic compounds. So, it is the basis of life on earth. Ultimately, all living organisms depends on the sunlight for energy. Yes or no? We used to get the food from the plants. Ultimate energy source is nothing but sunlight. So all the living organisms, for purpose of energy, we are depending on the sun. Sun is the main sources of energy for all the living beings. Now, importance of the photosynthesis. It is the primary source of food on the earth. So it is a main source or the primary source for all the living beings. So it releases oxygen to the atmosphere. Even in oxygen, if there is no oxygen, no organisms are able to live on the earth. If you tight your nose and your mouth tightly, even we cannot stay without air at least 1 minute. It is very hard to stay. That means it is oxygen which is released by the plants is also one of the most important component for the survival of a life. So oxygen is the byproduct of photosynthesis. So in order to study about the process of photosynthesis, we have some related experiments of the photosynthesis. So first is a variegated leaf experiment. Take variegated leaf. Variegated leaf means, you see the picture. combination of green and white color, green and pale yellow, you can see that, no, such a different colors of a leaf is called as a variegated leaf. So that was exposed to the light. We have to expose this to the sunlight, test the leaves for a starch. Then we have to test the leaf for the starch. So it shows the photosynthesis occurs. Only green part of the leaf in the presence of a light. How we will test for the starch? When we add the iodine solution on it, it turns into a blue-black in color. Where the part is turns into the blue-black color. In variegated leaf, we will see the white color and green color, no. When we pour iodine solution on a white color, we cannot find any change of a blue-green color. But when we pour the iodine solution on a green color, it converts into a blue-green in color. So that is nothing but conversion, presence of a starch. Iodine is an indicator which makes us to know that whether the starch is present or not. If the starch is there, it able to change into the blue-black in color. Okay, so that is the main function of the iodine. Next one is half-leaf experiment. That is, carbon dioxide is needed. How the carbon dioxide is needed for the process of photosynthesis. A part of a leaf is enclosed in a test tube containing KOH solution. The KOH is soaked into the cotton balls and keep into the bottle. You can see there in the picture. So the other half of a leaf is exposed to the air. Place this step in the light for some time. So test the leaf for a presence of a starch. Exposed part shows the positive starch and the portion in the tube shows negative. This proves that the carbon dioxide is required for the photosynthesis. See children, when you take a cotton ball and soaked into the KOH, what is the KOH capacity? KOH has the calibrity of the absorption of a carbon dioxide. KOH absorbs the carbon dioxide. If there is no carbon dioxide, the carbohydrates will not be produced or the process of photosynthesis will not take place. So the leaf which is dipped into the bottle into the KOH solution, the carbon dioxide is absorbed by the KOH, the process of photosynthesis is not takes place. And the leaf, half leaf which exposes to the sunlight, it is having sunlight as well as carbon dioxide, it could able to make the process of photosynthesis. You can see how we can make the process of experiment of presence of carbon dioxide for the process of photosynthesis. So when you plug the leaf which is kept in the bottle, the half which is in the bottle, when it is added with the iodine solution, it doesn't turn into a blue-black. But the part which is exposed to the sunlight, when we pour onto that part of iodine solution, it shows that change of a blue-black in color of the green. So we can say that we can define that the part which is exposed to the sunlight, it is participating to the process of photosynthesis, and the part which is inserted into the bottle, as the bottle is consisting of KOH solution, the carbon dioxide which is present in the leaf is absorbed by the KOH, and the leaf couldn't able to maintain the process of photosynthesis. That is about the process of carbon dioxide. Next one is experiment by Joseph Priestley. He said about the oxygen is needed for the life, not for the photosynthesis. Oxygen is needed for the life. He placed a mint plant in a bell jar. You can see the bell jar, mint plant. He found that the mouse stayed alive and the candle continued to burn. He hypothesized that the plants restore to their air whatever breathing animals and burning candles remove. So see children, he took a bell jar and he kept the candle and rat. The candle and rat are present only a bell jar, they die. But later he inserted the mint plant. When he inserted the mint plant, the carbon dioxide which is released by the rat helps to maintain the process of photosynthesis for the plant, and the oxygen which is liberated by the plant helps to the survival of the rat as well as burning of the candle. So it proves that during the process of photosynthesis, the plants used to release the oxygen. So the Priestley experiment make us to understand that the oxygen will be liberated during the process of photosynthesis by the plants. This oxygen helps to combustion of the candle as well as the life of the rat. and the carbon dioxide liberated by the rat is made use by the mint plant for the process of photosynthesis. This is the experiment given by the Priestley. And the next experiment is John Ingenhousz. Let us see about the John Ingenhousz. So, John Ingenhousz, he make experiment in the years of 1730 to 1799. He conduct the same experiment by placing it once in the dark and once in a sunlight. So, he showed that the sunlight is essential to the plants for the purifying the air, and he repeated the experiment with the aquatic plants. So, it showed that in a bright sunlight, a small bubbles were formed around the parts, while the dark they did not. And that means during the sunlight, the plants used to release the bubbles that are oxygen bubbles. But these bubbles are not released during the night time. That which is giving a result of during the sunlight only the process of photosynthesis is taking place by the plants. Later he identified these bubbles to be of oxygen. Thus he showed that only the green part of a plant release the oxygen. Only the green part of the plant are capable to release the oxygen during the process of photosynthesis. Next one is experiment by Julius von Sachs. He proved that glucose is produced when plants grow, and it is usually stored as in the form of a starch. And chlorophyll is located in a special bodies, and glucose is made in the green parts of the plant. So chlorophyll is made in the green part. It is a green part of the plant which mainly participate to process of photosynthesis. So by the middle of 19th century, it is observed that the plants use light energy to make carbohydrates from carbon dioxide and water. So that means mainly carbon dioxide and water. So empirical equation of a process of photosynthesis, you can see that CO2 plus H2O gives rise in the presence of light, CH2O plus O2. So where CH2O is represents here carbohydrates, that is nothing but a glucose, which is the form of a glucose. So experiment by Cornelius Van Neel. So Van Neel, CB Van Neel we used to call, no? Microbiologists conducted some studies in the purple and green bacteria. So purple and green bacteria. He demonstrated that photosynthesis is a light dependent reaction in which hydrogen from suitable oxides compound reduces carbon dioxide to carbohydrates. So carbon dioxide is converting into a carbohydrates. So these can be expressed by 2H2A plus CO2 gives rise in the presence of light, 2A plus CH2O plus H2O. This is the equation given by the Van Neel, CB Van Neel. So experiment by Neel, in a plants, H2O is the hydrogen donor and its oxides are oxygen. So purple and green sulfur bacteria used to H2S, green sulfur bacteria, as a H donor. So the oxidation product is sulfur or a sulfate, and no oxygen is produced. So thus he inferred that oxygen evolved by the green plants comes from the H2O, not from CO2. So this was later proved using radioisotopic techniques. So therefore overall correction equation for the photosynthesis is later, after the studies many taking place, the complete equation is given for the photosynthesis, important for your exam, that is 6CO2 plus 12H2O gives rise in the presence of light. You can write there chlorophyll also. So C6H12O6 plus 6H2O plus 6O2. So oxygen is a byproduct during the process of photosynthesis. So where does the process of photosynthesis will take place in the plants? So the plants consist of the special cells which are called as chloroplast cells. These chloroplast cells, they used to play a major role or a key role to maintain the process of photosynthesis. So chloroplast, photosynthesis takes place in green leaves and other green parts of the plant. So chloroplast present in the walls of the mesophyll cells of a leaves. So it helps to get the optimum quantity of incident light. Whatever the amount of light is needed, the chloroplast cell is able to absorb the light. Chloroplast contains a membrane system. It consists of grana, a membrane structure. You can see in the picture the word grana, that is a membrane like a structure, and stroma lamella and a fluid stroma. It has a stroma lamella and a fluid stroma. So each granum is a group of membrane bound sacs called thylakoids. You can see the sacs, no, like this. The steps like a structures. Each step is a sac. Many stacks are called as a stacks. These stacks together form as a granum. So each granum is a group of membrane bound sacs called thylakoids. Each one is called as a thylakoid. So they contain leaf pigment. They have the leaf pigment that is called as a chlorophyll. Okay. So chloroplast. The membrane synthesis trap light energy and synthesize ATP and NADPH. So it is called as light reaction. So production of ATP and NADPH. When the light is absorbed, what the materials are produced, ATP and NADPH. So in stroma, enzymatic reaction incorporates carbon dioxide into the plants for synthesizing sugar, which is in turn forms starch. It is called as dark reaction. So it does not mean that they occur in darkness or that they are not in a light dependent. So dark reaction means it occurs only in the dark. It means that either in the presence of light or in the absence of a light it may take place. But the light reaction compulsory will take place in the presence of the light. Okay. So the photosynthesis site. So pigments involved in the process of photosynthesis. Pigments are substances that have ability to absorb light at a specific wavelength. So chromatography shows the following leaf pigments in the chromatogram. So that is chlorophyll A, bright or a blue green. Chlorophyll B, yellow green. Xanthophyll, it is in yellow color. And carotenoids, yellow to yellow green. So different colors are given for every pigment. That is chlorophyll A, bright or a blue green. Chlorophyll B, yellow green. Xanthophyll, yellow. And carotenoids, yellow to yellow green. So these are the different colors. Chlorophyll B, xanthophyll and carotenoids are accessory pigments. Chlorophyll B, that means main chlorophyll is a chlorophyll A. Chlorophyll B, xanthophylls and carotenoids are the accessory pigments. So functions of accessory pigments, what they will do? They absorb light at the different wavelength and transfer energy to a chlorophyll. They absorb the light and it will be transferred to the chlorophyll. They protect the chlorophyll A from the photooxidation. So they protect the chlorophyll in the process of photooxidation. So the absorption of spectrum, action of spectrum, considered this is waves, the length of a chlorophyll, how much chlorophyll has to be taken. So this is not much need for you, so no need. So photosynthesis pigments are organized into two photosystems called photosystem 1, PS1, photosystem 2, PS2. These are named as the sequence of their discovery. So each photosynthesis has a chlorophyll A and a axis. Every system has the chlorophyll A and accessory pigments bound by the proteins. They will bound by the proteins. All pigments except one molecule of chlorophyll A from light harvesting complex, that is a grana. Light harvesting complex is a grana. Single chlorophyll acts as a reaction of center of PS1, that is a light reaction. The reaction center absorbs light at a 700 nanometers and called as P700. So in phase 2, the reaction centers absorb the light at 680 nanometers and so called as a 680, P680. Now the next one is a photochemical reaction. So, light reaction includes light absorption, water splitting, oxygen releasing, formation of ATP and NADP. So, ATP and NADP are high energy chemical intermediates. What are they? High energy intermediates. And ATP we can call it as a adenosine triphosphate. NADP means nicotinamide nicotinamine adenosine dihydrogen phosphate. So ATP we call it as a energy currency because the energy is stored in the form of a phosphate molecules. Okay, so the light reaction consists these steps: light absorption, water splitting, oxygen release, formation of ATP and NADP. 4 steps are involved in the light reaction. So the electron transportation. When phase 2 absorb red light of a 6-8 nm wavelength, electrons are excited and transferred to an electron acceptor. So the electron acceptor passes them to a chain of electron transport system consisting of cytochromes. So this movement of electrons is downhill in the terms of redox potential scales. So these electrons are transferred to the pigment of PS1 stage. Next one is a splitting of the water. So the first step, electrons means the light breaks when the light is absorbed, electrons will be break down and the energy will be released. So there in the process, when the absorption of light takes place, electrons will be split and it is released into the water molecules. So here we can see the breaking down of water molecules. The water splits complex in a PS2 is located on the inner side of the thylakoid membrane, inside the thylakoids. So a group of thylakoids form the granum, no? So inside the thylakoids, water is split into H plus and O and electrons. H2O gives rise 4H plus O2 plus 4 electrons. Okay. So PS2 can supply electrons continuously by replacing electrons from the water splitting. So phase 2 provides the electron needed to replace the removal of PS1. So the protons, H plus ions, are used to reduce the NADP to it will be reduced into NADPH. So oxygen is liberated as a byproduct of the photosynthesis, even as also in the light reaction. So photophosphorylation. The synthesis of ATP by cells in a mitochondria and chloroplast is called as a photophosphorylation. That means development of ATP. Formation of ATP molecule in the mitochondria and the chloroplast is called as the photophosphorylation. This is not neat, just photophosphorylation is the synthesis of ATP and ADP. So in chloroplast, in presence of light. So it occurs in 2 ways, non-cyclic and cyclic processes. So cyclic photophosphorylation, it occurs in the stroma lamellae in dark reaction. So when phase 1 is functional, the electron is circulated with the photosystem and ATP synthesis occurs due to the cyclic flow of electron. So the lamellae of a granum have a PS1 and PS2. So the stoma lamella membranes lacks the PS2 and NADP products. Okay, the electron does not passes on to the NADP, but the cycle back PS1 through electron transportation. Here only ATP is the synthesized, no NADP or a H plus. So cyclic photophosphorylation is also occurs only when light of a wavelength beyond the 680 nanometers are available. So now the next one is the photochemical reaction follows with the dark reaction. Dark reaction is also called as a biosynthetic phase. So products of a light reaction are ATP, NADPH and O2. So dark reaction is the use of ATP and NADPH. So in dark reaction, ATP and NADPH will be used to derive the process of for the synthesizing of the sugars. Carbohydrates will derived in the dark reaction. So this phase does not directly depend on the light, but it is dependent on the products of the light reaction. So it can be verified as follows. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. So if light is available, the synthesis starts again. So when the light is slow down, the dark reaction will start and it will be stopped. Again when the light comes, it starts to make the dark reaction. So the synthetic reaction will be started. So carbon dioxide combines with the water molecule and C2H4 or sugars. So carbon dioxide assimilation during the photosynthesis is 2 types, that is C3 pathway Okay, so in this first product of carbon dioxide fixation in a C3 organic compounds, that is a 3-phosphoglyceric acid forms PGA, 3-PGA. So this is a Melvin Calvin discovered. This process is covered by the Melvin Calvin. That's why it is also called as a Calvin cycle. Okay, discovered this using of 14C in a algal photosynthesis. So in these first stable products, oxalic acid and OAA and 4 carbons C4 acid will be formed. So what are the in Calvin cycle in dark reaction? What does go in come out of the Calvin cycle? So 6 carbons will be accepted by 6 carbon molecules, carbon dioxide molecules are accepted. Due to that, it will be released in the form of a 1 glucose molecule. 18 ATP molecules are accepted and released out 18 ADP molecules. 12 ADPH molecules are taken and 12 NADP molecules will be released out. So no need of pathways. Now up to now we discussed about how the process of photosynthesis will take place. Now we will discuss that factors affecting photosynthesis. How the factors will affect the process of photosynthesis. So internal plant factors. So the size, age and orientation of our leaves, mesophyll cells, chloroplast, carbon dioxide concentration. These all are the internal factors which cause the process of photosynthesis, and the external factors are sunlight, temperature, carbon dioxide concentration, water. So plant factors are dependent on the genetic predisposal and disposalization and the growth of the plants. These factors depends on the organs. So now let us see Blackman's law of limiting light factors. No need of carbon dioxide concentration. Major limiting of factor of photosynthesis. Carbon dioxide concentration is very low in atmosphere. 0.03 to 0.04% in the atmosphere. In case of up to 0.05% causes increase in carbon dioxide fixation rates. Beyond these levels, it becomes as damaging over longer periods. So at low light conditions, C3 and C4 plants do not respond to the light. So carbon dioxide condition at a high light intensity, they shows increase the rate of the photosynthesis. If the more amount of light is there, it increases the rate of the condition of photosynthesis. How the temperature affects the process of photosynthesis. Dark reaction begins enzymatic or temperature controlled. Influence of the temperature or a light reaction is very less. The C4 plants responding to the higher temperatures and shows higher rate of photosynthesis in C4 plants when there is more temperature. C3 plants have much lower temperature optimum. The temperature optimum of plants also depends on the habit. Tropical plants have high temperature optimum than the plants adapted to the temperature climatic conditions. Depending on the temperature also, the condition of photosynthesis takes place. Water, coming to the water stress closes the stomata, hence reduces the carbon dioxide availability. That means when there is less amount of water, the stomata will be closed. We can see the transpiration rate will be reduced. So water stress also with wills leaves. So when there is no water, the water stress wills the leaves. That means enfolded the leaves. Thus reduce the surface area of the leaves and their metabolic activities. When there is a water is reduced, the metabolic activities also will be reduced. So then how the process of photosynthesis is maintaining of affecting different types of factors, how the plants could able to prepare the food substances. These all are the photosynthesis is the process of autotrophic nutrition. So up to this we discussed about the process of autotrophic nutrition. So if it is available a small activity, if you are able to collect different types of cuscuta plants, try to collect or study how the cuscuta plants are entering into the host plants and how they are sucking the nutrition from the host. Try to observe this from your surroundings, children. Hope you understood this is autotrophic nutrition. We shall meet into the next session. discussing about the human nutrition. Thank you, children.